Melvina.
I'm Fatih. I'm Imam.
And we have Fico. And today we will be doing the cell membrane bubble wrap experiment. Okay, for the ingredients we have the J-loop. For in this case we have the bubble. We have 4 bendable straws, 1 clean straws, spool of cotton thread, and shallow trays. We will be connecting the 4 bendable straws into a square.
This is the final shape after reconnecting the straw.
Membranes are fluid and flexible. Notice how the bubble film is able to bounce up and down without breaking. This is similar to the cell membrane. You can also see the light reflects on the bubble film. This demonstrates how the molecule move on the cell membrane. Concept 1 was able to show us how membranes are fluid and flexible. They are not static and change to be able to adapt to changing conditions that the cell membrane undergoes.
Cell concept 2. Membranes can self-repair. As you can see, even with the whole hand going into the film never breaks, and when the hand is taken out of the film, it is able to go back to the same shape it was before. This is similar to the cell membrane because it is able to repair after there is breaks in the phospholipid bilayer. Concept 2 was able to show us that the lipid bilayer can be fixed by the attraction of the phospholipids in the cell membrane.
Cell concept number 3. Eukaryotic cells feature membrane-bound organelles as a smaller bubble is formed inside a bigger bubble. This shows that layers are created due to a creation of a separate air compartment. In conclusion, we are able to see that in a eukaryotic cell, the membrane also has phospholipid layer and they have membrane-bound organelles that create compartment within the cell. The primary structure of outer cell membrane as well as the membranes that enclose organelles is a double layer of phospholipids known as the phospholipid layer.
Now, cell concept 4. Membrane proteins perform special functions. This idea consists of a formation and existence of specialist proteins which are embedded within the lipid bilayer which give the membranes unique properties. One example of these proteins are channel proteins. These proteins form a passageway for larger or electrically charged molecule to pass through the membrane. Just like the pencil made the thread expand and move. These proteins are also able to drift through the lipid bilayer in the cell membrane.
Gap junction egg transport between animal cells. In this concept, bubble was formed and then the straw was lifted out of the bubble, forming an airway. This explains the idea that gap junction form between neighboring animal cells, allowing their cytoplasm to connect directly, therefore provides a quick way to move material between two cells.
Cell concept 6. Many bacterial cells reproduce through binary fission. In this step, a bubble was formed and a thread was put into the water and lifted through the middle of the bubble. The bubble rapidly split into 2. This concept relates to the idea of binary fission, which is how some single-celled bacteria reproduce splitting into 2.
So that is it for our cell membrane bubble wrap experiment. Thank you for watching.